Thus far in evolution, we've been studying scientists that contributed to the theory of evolution. For example, Darwin, Lamarck, Cuvier, Lyle Hutton, and more. But we've also been focusing on natural selection, which is a mechanism that leads to evolution. And so far, emphasis has been on a special type of evolution called microevolution, which is a change in allele frequencies over time. So, for example, if you look at this generation over here, we can see that for color genes, 80% of the alleles are green and 20% are brown. But after 1 year, we see a noticeable change. We see that 60% are green alleles and 40% are brown alleles. So the green went down and the brown went up. So maybe a mechanism for this change may have been natural selection, where perhaps the green bugs were being eaten, couldn't survive, couldn't reproduce as much, and the brown bugs were somehow camouflaging, hiding, could survive, could pass on their genes, and so they were increasing. And so if you study this population for another generation, you might see more changes. But this is microevolution in a nutshell. And so the evolution piece comes from the fact that the gene frequencies have changed from one generation to another. And the natural selection piece comes from the fact that the mechanism that led to that change, the cause of that change, was maybe some kind of differential reproductive success caused by different fitness in the environment. But so far, you've seen here that I've assumed that it was natural selection that caused this because we only learned one mechanism. But in reality, there's more than just one mechanism that can lead to evolution. It's not just natural selection that can lead to this change in gene frequencies over time. So the purpose of this PowerPoint is to explore what type of mechanisms can cause a population to evolve or to microevolve, kind of like natural selection. We're going to see natural selection in a bit more detail in this presentation as well, but we'll also see other mechanisms responsible for evolution. So a few terms we need to review before we continue through this presentation. This is something that you've seen in ecology. This is called the hierarchy of biological organization. So when you studied ecology in grade 9, you learned that you have individuals. The individual organism is one of that particular organism. But sometimes you have many of those individuals living in the same environment, in the same habitat, and those are called populations. So a population is many individuals of the same species living in the same environment. So you might have many butterflies of the same species living together, many deer of the same species living together in one environment, many bears living in one environment. So many bears together would be a population of bears, many butterflies together would be a population of butterflies. So when we talk about populations on a evolution level, a biological level, it has to be organisms of the same species. Now, you don't really know the definition of species yet. You can look it up. We're going to talk about it in more detail later on, but basically the same type of organisms. It's more complex, but we'll get to it later on. Remember that in populations there exist variations, and the variations occur at the gene levels, and as a result we see them at the phenotypic level oftentimes. So we're going to go back and just backtrack a bit to take a look at where variations come from and see a few other terms and how they connect to evolution. So think back to genetics now. So what I want you to understand in biology is that many of the concepts that we study are connected together. In fact, all the concepts we study are connected in some way, shape, or form. And the more you can realize those connections, the more you truly understand the biology that we've been studying. So here we're going to see how genetics is connected to evolution. So when we study evolution, remember, it's a change in allele frequencies, gene frequencies, more specifically allele, I should say, over time. And so there is a direct connection. It's a change in the allele frequencies. And we studied alleles in genetics. So when we study evolution, we look at something called the gene pool, and that's all of the alleles in a given population. So we usually study, at least in grade 11, one gene at a time. So here we have the B gene, and the B gene has two alleles. There's a big B allele and a little b allele. And so if we're studying the gene pool, we could say that there's some little b's in this gene pool and some big B's in this gene pool as well. And you could actually, if you did some genetic testing, if you studied phenotypes, you can take a look and determine which allele is more frequent, the little b1 or the big B1. And there's actually ways to do that. We'll see later on. Remember that the genotype is the genetic composition, so what alleles you have. So this individual has big B, this individual has little b, this individual has big B, little b, heterozygous, homozygous dominant, and homozygous recessive. And remember that the genotype is connected to the phenotype. Essentially, the genotype is the DNA code that you have, and the phenotype is the expression of that. Is what it ends up looking like in the organism. It might be an outward appearance, a physical appearance that you can directly see, or something more like blood type. But the idea is that maybe this big B gave the individual a certain type of fur compared to the little b that would give the individual a different type of fur. And you can take a look at this picture over here as well. These butterflies over here, there's different alleles, and as a result of having different alleles and different combinations of alleles, we have different phenotypes. So for example, you can see if you have little a, you have this white coloration. If you have big A, you have this brown coloration. And if you have big A, you have this orange coloration. And hopefully it's not too far back. You can think about what type of dominance this is. Perhaps it looks like an incomplete dominance, maybe, right? Because we have an intermediate phenotype between the two, and we don't see the brown and the white, but we see something kind of in between those colors there. So again, the genotype determines the phenotype. And remember that there's variation. So because there's different types of alleles, different combinations of alleles, the individuals will differ in their phenotypes. And so far we've seen that in order for natural selection to act, you need to have differences in the population for natural selection to act on them. But those differences arise directly from the genotype, and they lead to the phenotype. So let's go back and take a look at the sources of variation. So the main idea of the previous slide is that there's gene pool. There's different alleles in the populations, and based on different genotypes, there are different phenotypes, there's variation. But where do those variations come from? Well, why are there so many different variations in populations? So what we've already seen is that the original or ultimate source of all variation are mutations. So a random change in DNA sequences, a change in the letter A, T, C, G, in that code that we saw earlier, can lead to new alleles and perhaps new characteristics in the population. Not all the time, but many times it can. So we can take a look over here. Here we have some green insects, and they have two alleles for the color gene. So let's say they have little b, both of them. And none of them have any other alleles for color, it's just pure green there. But you notice that they have kids, and most of their kids are green, but there's one offspring that ends up having a brown coloration, and you'll notice here that there's a difference in one of the DNA molecules, that there might have been a mutation that occurred, and that led to some dominant allele. And the reason I know it's dominant is because when you have one copy of the brown allele and one copy of the green, the brown color is the one that shows up in the insect. But the idea here is that where there was no variation before, where there was only green, variation arose through a random mutation that occurred. It wasn't caused on purpose. The insects didn't say, I really want a brown baby, so I'm going to cause a mutation in my DNA. It just happened randomly through mistakes in DNA replication. Remember, we saw S phase, where DNA has to replicate. If there's mistakes there, there's a mutation that can happen in the gametes, and then you have, a baby with maybe a different phenotype. It's a possibility. So now, this mutation led to variation within the population, and perhaps, if this is heritable variation, it can be passed on to the next generation. So, a few things I want you to understand about mutations is that they often have a bad reputation. We often think they're always bad, and they're always going to put the organism at a disadvantage. But it's not always the case. We're going to start off by taking a look at bad mutations. So mutations that give individuals a disadvantage, that basically reduce their fitness and reproductive success, they're called deleterious mutations. So here we can see a picture of a peacock, and you'll notice that it's very, very white. If you've ever seen a picture of a peacock before, they should be very, very colorful if they're male peacocks. The females aren't colorful, the peahens, but the males are colorful, and that's because it allows them to attract the females so that they could survive, so that they could reproduce and pass on their genes. Now, this coloration is caused by a mutation. You'll notice it's a homozygous recessive mutation. And again, that's why I like to start off the course with genetics, because a lot of the terms in evolution refer back to genetic terminology. So you can see here that this recessive mutation led to essentially lack of coloration. And so this is a deleterious mutation for this organism, because now that it's all white, the peahens won't really dig it anymore. They don't really want to go and reproduce with this white peacock over here. Peahens are kind of picky. So this individual will not pass on its genes anymore, will have a reduced ability to do so, because it's not going to really have mates seeking it out like it would if it was very, very colorful. So that's one example of a deleterious mutation. It doesn't seem like it's bad. It's just, it still looks kind of pretty, but it's not good enough for the peahens. Other examples of deleterious mutations would be mutations that might cause disease in humans and other organisms. But like I said, not all mutations are bad. Some can be good, so we can have beneficial mutations. Beneficial mutations are the opposite of deleterious. They give you a increased fitness, increased reproductive success in your particular environment. So some bacteria, they will sometimes have mutations that make them resistant to antibiotics. And so a lot of times we wonder, well, why is this beneficial? I mean, isn't that bad? Don't we want to kill bacteria? Well, the point is that it's beneficial for the bacteria. When we look at mutations, we look at it in terms of the organism that it happens in. So for bacteria, these mutations are beneficial for them if they have antibiotic resistance and there's antibiotic present in the environment. There might be some other examples you can think of. Would be, there's some mutations that protect you from certain diseases. So there's some mutations that maybe protect you from HIV. That could be an advantage if that virus ever became more widespread. So that could be considered a beneficial mutation. Or maybe you might have some genes that reduce your chance of getting cancer. And so if that allows you to survive more, reproduce, pass on your genes, that could be a beneficial mutation. And lastly, many mutations are just neutral. There's a lot of mutations happening in you as you listen to this, as you're studying, but they're not really having any effect on your phenotype, at least visibly, and they're not really reducing or harming you in any way in terms of reproductive success. One great example of that is heterochromia. Heterochromia is a result of a mutation, and it gives you two different color eyes. And these people they're not harmed by it. It doesn't hurt them. It's not a disease, and they don't really benefit from it. So they don't really have increased fitness as a result of it. Now, one day, if, for example, everybody started to become attracted to people with heterochromia and they all want to reproduce with people with heterochromia, then in that case, that mutation may go from being called neutral to being called beneficial. So it all depends on the environment and the people that you're surrounded by as well. So that's the key theme. It all depends on the environment. A mutation is not necessarily always deleterious or always beneficial or always neutral. It always depends on the context. So for antibiotic resistance, for example, if there's no antibiotics present, well, it doesn't give the bacteria an advantage, so it's actually a neutral mutation. The effect is neutral. Now, if there's antibiotics present, then it's beneficial. What's interesting is many times when bacteria get resistance to antibiotics, it can often be deleterious. Sometimes the resistant bacteria, they'll actually reproduce slower than the non-resistant one. And so if antibiotics are not present, it doesn't really give them an advantage. It actually hurts them a little bit. So it all depends on that context again. So make sure you remember that when you study mutations. They're not all bad, they're not all good, they're not always all bad, they're not always all good, and they're not always neutral. It depends where they're in. So mutations were the original source of variation. They give us some raw variety materials to work with, but remember from genetics we saw there's other ways to lead to variation. So in sexually reproducing organisms we have meiosis, and so meiosis has a few different processes that shuffle those genes around, those various genes around to get different combinations. So we had independent assortment and crossing over that can lead to a great deal of combination of gametes. Something that we didn't study yet, but we will eventually. There's some organisms that we studied initially that study asexually. So, for example, bacteria, they study asexually. They don't have meiosis, they don't have gametes, and they produce these babies by splitting, and they're identical to each other. And so you might think, well, the only source of variation for them is mutation. They don't have meiosis. So they can't really do independent assortment and crossing over. They're making clones of themselves. So they're relying mostly on mutations, which is true. But another cool thing that bacteria can do, and this does not count as sexual reproduction, it just counts as something called recombination, is sometimes bacteria can connect to each other by this thing called a mating bridge, and they can kind of exchange DNA with each other and lead to different combinations in bacteria. So you might have a resistant bacteria here and a non-resistant bacteria over here, and it can pass on its DNA to the non-resistant one and make it resistant. So even in asexually reproducing organisms, there are some other mechanisms that are available to shuffle genes around. This particular term here is called conjugation. And we'll see other examples in bacteria later on. It's not sexual reproduction, it's more recombination. The reason it's not sexual reproduction is because it's not making more individuals, it's just shuffling some genes around. So just understand that mutations are are quite responsible for the variations in asexual organisms, but there are other mechanisms as well. But anyways, back to meiosis, that was an aside. Back to meiosis, independent assortment crossing over lead to variation in the gametes. And then as a result of having many different gametes, you can have random fertilization. So the different gametes can combine in different ways, and that leads to even more variety of offspring in the population of organisms that you're studying. And so that's something we saw in the first unit. If you don't quite remember that, you can go back and study it in the meiosis PowerPoints. So we saw sources of variation. You need variation in order for evolution to actually happen. So let's go and take a look at our gene pools again. Let's understand gene pools in a bit more detail. Like I said, microevolution is a change in allele frequencies. So to know if there's a change in allele frequencies, you need to know what your population starts off as, what it looks like to begin with, to see if it ends up changing in the end. So we can actually study gene frequencies. Let's look at flower color here. And I should more specifically say allele frequencies, right? So the frequency of the different alleles for a given gene. So here we have the flower color gene. There's two alleles, big R and also little r alleles. Now in this population, if you want to calculate the frequency of the dominant allele, so let's call that big R, the frequency would be, put your total number of alleles at the bottom. There's 20 alleles total. How did I know that? Well, you can go and count all the different letters. 1, 2, 3, 4, 5, 6. You could do that. Or you could count the individuals. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And multiply by 2, right? Because each individual has 2 alleles for a gene. And then go count. Now you have to go count your big R's. So your big R's, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. You have 14 big R's, so that gives you a frequency of 0.7, or sometimes you might write it as 70%. Now, you can also find the frequency of the little r. There's a few ways you can do that. First of all, realize if big R is 70%, little r has to be 30. You just do 100 minus 70, and that gives you 30, or you do 1 minus 0.7, and that gives you 0.3. So you could do that, but just in case you want to actually check your work, you do the same thing, count the total number of individuals, multiply by 2, and then count the number of little r's you have. 1, 2, 3, 4, 5, 6. 6 little r's, that's 0.3 or 30%. So you can see that you have a higher frequency of dominant alleles in this case than recessive alleles. It's not just because it's dominant that it's higher frequency, it just happens to be this way. Maybe the environment was better for the red than it was for the white. But it's possible in some populations you have more recessive than dominant. That's a possibility as well. So let's take a look at one more important thing I want to discuss in this slide here is that these letters will change. Some students might use big A for red, little a for red. So usually in evolution, when we study it, we give the dominant allele the letter P. P represents the dominant allele. So if I were to write this down properly, I'd say the frequency of the dominant allele is 0.7. And this is P, frequency of dominant allele. The recessive allele, we give it the letter Q. And that's going to be 0.3. So P represents big R in this case, and Q represents little r in this case. So just to be consistent, we give P for dominant, Q for recessive. And there's a really cool relationship I need you to remember all the time. P plus Q will always be 1. So 0.7 plus 0.3 will equal to 1. Basically means 70% plus 30% will be 100. So if you know one percentage, you can figure out the percentage of the other by doing 100 minus the percentage of the other. So for example, if you know P, you can find Q by doing 1 minus P. If you know Q, you can find P by doing 1 minus Q. And so you should be able to calculate frequencies for populations that are given to you in picture form or in paragraph form. I'll walk you through a few examples, but I want you to try these on your own as well. Based on what we saw in the previous slide, you should be able to do something like this. So the first question is calculate the frequency of the dominant allele. I'm not going to count the alleles in the water here. This is just a picture to show a gene pool. I'm just going to calculate the alleles in the individuals, the population over here right now. So let's take a look. So for the individuals, I have, let's count the dominant allele, so we'll call that P. There are 1, 2, 3, 4, 5, 6, 7 individuals, so that's going to be 14 alleles. And the big B, we have 1, 2, 3, 4, 5, 6, 7. So our P is going to be 0.5 or 50%. So that's done over here. Our recessive allele, we can just do 1 minus P to give us our Q, which is a recessive allele. And that would be 1 minus 0.5, which is 0.5. So we have P and Q are equal to each other, and if you check at the end, 0.5 plus 0.5 add up to 1. P plus Q is equal to 1, right? Remember that little trick there? P plus Q is equal to 1. Next is calculate the proportion of homozygous dominant individuals in the population. So that just means find the decimal or the percentage value. So the homozygous dominant individuals, there's 1, there's 2, nope, that's heterozygous, sorry. 1, 2, I think that's it. So 2 out of, not 14 this time, because it's the number of individuals that are homozygous dominant. So here we're calculating the allele frequency. Here we're calculating the number of individuals, and there's only 7. So 2 out of 7 would be 2 divided by 7. Looks like about 28.57%. You could have also kept it as a decimal. Proportion of homozygous recessive individuals, so 1, 2, looks like there's 2 there. 2 out of 7. Also, sorry, I should multiply it by 100 if I'm going to write that down there to be mathematically correct. Multiplied by 100%. If you don't do that, you technically shouldn't write it down as a percentage because you didn't mathematically show you did that. 2 over 7 times 100%, and that gives us our 28.57. And again, you could always just figure out the last one by subtracting these two values from 100, but I'm just going to go and make sure that it makes sense in the end. So my heterozygous individuals, I have 1, 2, and 3. So 3 out of 7 times 100%. And we get 42.85. If you add them all up, you'll get pretty close to 100%. You might be a little bit off just because of the rounding that you might have done, but that's essentially how you go about doing these type of problems. So you should be able to do problems like this now. where you get a picture and you're able to reason through questions like this. So I would recommend that you pause the video and then compare to my answers afterwards. I'm going to write the answers. I'm not going to do a thorough explanation of them, but I do want you to compare and see if you can do this on your own. So our first question is, which allele is dominant, green or red? And so you have to analyze the individuals, and what you'll notice is you have a green individual over here with both alleles, a green and a red. Yet the color that shows up is the green color, so because of that I know that the green is dominant, since an individual with both alleles ends up being green, so green kind of wins and takes over. Calculate the dominant allele frequency, so they're asking us for P over here. There are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 alleles, so there's 20 alleles total. How many green alleles are there? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 out of 20, which is 0.6 or 60%. We can figure out the recessive allele by doing 1 minus 0.6, which is 0.4, 40%. So there's a higher frequency of green alleles than there are red alleles. Calculate the proportion of homozygous dominant individuals. So let's go calculate our proportion here. So homozygous dominant individuals, we have 1, 2, 3, 4. 4 out of 10. And you multiply that by 100%, or you can leave it as a decimal form. That's 40%, our homozygous dominant. I'm not going to show the work for the last two, I'm just going to explain it and then write the answer down. Recessive individuals, so homozygous recessive individuals, or, well, I didn't put homozygous because if they're recessive phenotype, then they have to be homozygous recessive in this case. So we have 2 out of 10. So that's 20%. We would expect our heterozygous to be 40% as well. Let's just go double check, see if that's true. 1, 2, 3, 4. 4 out of 10 heterozygous individuals, so 40%. So this is our p-value, our q-value, and then our different number of individuals in the population just using the images. So there's a famous equation that you'll get to learn later on. It's called the Hardy-Weinberg equation, and it relates to something called the Hardy-Weinberg equilibrium developed by some scientists. What they said in their concept is that if you have a population and its allele frequencies are not changing, then the population is not evolving. Okay, so if a population is in Hardy-Weinberg equilibrium, the population is not evolving, meaning the allele frequencies P and Q are staying the same from generation to generation to generation. There might be a slight fluctuation. You do statistical tests to figure out, what's acceptable. It's a slight fluctuation, but overall, there's no change from generation to generation. So from generation 1 to generation 2, the R and the little r will be, the big R will be 0.7, the little r will be 0.3. So in order for a population to not evolve, in other words, to be in Hardy-Weinberg equilibrium, HWE, Hardy-Weinberg equilibrium, there are 5 conditions that need to be met for a population to keep the same allele frequencies. And now, hopefully you're starting to make the connection. The mechanisms of evolution are these 5 conditions not being met. So firstly, for a population not to evolve, there can't be natural selection. If there is, you'll be knocked out of Hardy-Weinberg equilibrium and the population will evolve. You know natural selection already, but we'll go into it in more detail later on. I'm going to define all of these as we go through them throughout the PowerPoint. There can't be gene flow. If there is gene flow, then the population may evolve and be out of Hardy-Weinberg equilibrium. There can't be mutations, because that could lead to evolution. There has to be random mating. If there's no random mating, then there may be evolution. And the populations have to be large, because if the populations are small, then evolution could happen. And I'll explain all that later on. But the main point is that if any of these conditions are not met, then evolution might happen. So for evolution not to happen with certainty, all of these conditions need to be checked off. There's no natural selection, there's no gene flow, there's no mutations, there's random mating, and the populations are large. You can fit all those on your 5 fingers on your hand. There's actually a video it's called the 5 Fingers of Evolution. It's on the course website. You can take a look at it, or you can look it up on YouTube. It's a great explanation of these different mechanisms, all summarized in a very short video, but very well done video as well. So essentially, if any of these conditions are not met, if they're violated, meaning not met, then the population is going to be out of Hardy-Weinberg equilibrium, and gene frequencies, or I should say allele frequencies to be more precise, allele frequencies may change, and that's evolution. Okay, so if your population has microevolved, it's not in Hardy-Weinberg equilibrium. If it has not evolved, then it is in Hardy-Weinberg equilibrium. Okay, so think equilibrium meaning stable, nothing's changing. So Hardy-Weinberg equilibrium, no evolution. Evolution, there's no Hardy-Weinberg equilibrium. So in your textbook, you can read up on this in more detail. There's also a great gizmo that you're going to go over to learn about this as well in more depth. There's a cool formula that you'll use to actually predict allele frequencies, or frequency of individuals based on allele frequencies, and that's called the Hardy-Weinberg equilibrium equation. So here we can see a change in allele frequencies over time. 75% green, 25% of this brown here, and then over time there's a decrease in green, increase in brown. So this population may not be in Hardy-Weinberg equilibrium. It's evolving, so it's not in Hardy-Weinberg equilibrium. If these percentages had stayed the same, then the population would have been in Hardy-Weinberg equilibrium. So again, you can see in this picture over here, we have our initial population. The allele frequencies have been calculated for you. Big A is 0.5, little a is 0.5. Here we have our population after a generation or so, and notice that the allele frequencies are different. The big A went down and the little a went up. So maybe there was natural selection. Maybe there was something called gene flow. Maybe there was something called genetic drift as a result of small population sizes. But the main point is that this population is not in Hardy-Weinberg equilibrium. It has a change in allele frequencies because of one of the mechanisms coming into play. And so we're going to see what those mechanisms are. So like I said, Hardy-Weinberg equilibrium means not evolving. If you are evolving, you're not in Hardy-Weinberg equilibrium, and it's due to something here not being in check. So these are the scientists that came up with the Hardy-Weinberg equilibrium concept. So again, when you read up on when you're reading your textbook, do take a look at their contributions and understand what they explain and how they came up with it. So let's start off with the first checkpoint on the Hardy-Weinberg equilibrium. So they were saying that for a population not to evolve, well, or to be in Hardy-Weinberg equilibrium, it's important that there's no natural selection. So if there is natural selection, then the population may evolve. The population may have a change in allele frequencies. And it just makes sense. Some individuals are being selected for and some are being selected against. So this bug here is being eaten more frequently than the brown ones. And so that's going to lead to a change in allele frequencies over time. And it just so happens that the reason the brown ones are not being eaten is because they're camouflaging on the brown tree over here. So the cool thing about natural selection is that it's a mechanism of evolution that leads to adaptations, that basically leads to organisms seeming like they're better fit for that environment because they were just better in that environment than the other organisms. So natural selection is a mechanism of evolutionary change. It can cause evolution. If natural selection does occur, the population will no longer be in Hardy-Weinberg equilibrium and microevolution can occur. So what we're adding though to this portion of natural selection here this time is natural selection is actually a few different patterns that you might see. Natural selection can be of three types. It can be stabilizing selection, directional selection, and disruptive selection. So we're going to take a look at what those three types are by studying some population graphs. Let's start with stabilizing selection. So I want you to notice that in this graph we have our original population highlighted. The curve shows the frequency of different phenotypes of individuals in the population. So you'll notice that individuals at the extremes, the very light one and the very dark ones, have a low frequency, and individuals in the middle have a high frequency. So let's say natural selection occurs. If natural selection occurs and it just happens to be what's called a stabilizing selection, what you're going to notice is that compared to your original population, the extremes are going to go down even more. So the extremes are selected against, and the ones that are selected for, well, they're the ones that were the intermediates, the in-between phenotypes. So you can see, so this dotted line represents the original population, and this dark line here represents the new population after natural selection. This is called stabilizing selection because the majority of the ones that survived were the ones with the in-between phenotypes, not the ones at the extremes, the very light ones or the very dark ones. So that's stabilizing selection, where the in-between ones are selected for over the extreme ones. Sometimes you might have what's called directional selection. So again, here's our original population. We have a variety, a range of these. Typically, the extremes are always kind of lower than the intermediates, unless there's, again, an environmental condition that leads to a change. But in a directional selection, what we notice is, take a look at your shift here. This is our original graph, our original population. The individual shifted more this way, towards the darker end of that spectrum. So in directional selection, there's a selection for one of the extremes. It could be for the, let's say, the very dark individual, or it could have gone this way. It could have gone to the very light individual. So it's for one of the extremes. So in directional selection, there's one particular direction that's chosen. It's either going to the very dark extreme or to the very light extreme. It doesn't just have to be fur color. There's other examples that we'll see later on, but it's going to one extreme or the other. So stabilizing was staying kind of in the middle. More of the middle ones survive, and directional is choosing a direction, either going to one extreme or the other extreme, not to both. And that's directional selection. Sometimes natural selection can be what's called disruptive. We also call this diversifying selection. So the interesting thing about this one here is that the intermediate ones are selected against this time, and the extremes are favored. So let's take a look at our original population over here. You can take a look here. The intermediate ones decreased, and the extremes, they were favored. And the reason this is not directional is because it's both extremes that were favored in this case. It's called disruptive because it looks like it disrupts your curve. It kind of comes in and smashes it down in the middle, so it disrupts it. And it really leads to to populations where they look very contrasting. You have some very, very light individuals and some very, very dark individuals only, and very few of the intermediate types. So very contrasting, distinct phenotypes that you'll see in the population as a result of disruptive selection. So these are all examples of natural selection. It's just we call it different things based on what was selected for and what was selected against. So if the intermediate middle guys were selected for, we say it's stabilizing. If one of the extremes was selected for, we say it's directional. And if both of the extremes were selected for, then we say it's disruptive. I do want to mention for directional that you still have that range that exists. It's just that there's a higher proportion closer to the darker side of things. So here we can see a summary of the different results of natural selection: directional, stabilizing, and disruptive. So you can take a look at the examples that are provided here. So you can see the long wiggly tails that look like snake and scare the predators. So that basically means that the lizards with longer tails are going to survive, reproduce, and pass on their genes. So initially you might have had this is your original population. The longer tail lizards were surviving because they could scare off predators, and so you have more longer tail lizards. So individuals with longer tails are favored, and we have some directional selection happening in the direction of the longer phenotype compared to the shorter tail phenotype. Sometimes you might have the intermediate that survives. So short tails mess up the cat's balance. Long tails drag on the ground. Medium tails are best. So the intermediate one is the one that was better because the long tails, they drag on the ground. The shorter tails, they're not good for the balance. So medium seem to work better. So we have a stabilizing selection that occurred. And over here we have some disruptive selection or diversifying selection. So short tails help keep predators from catching you on the ground. Long tails are good for balance in the trees. Medium tails, they don't really help at all. So if you have a short tail or a long tail, that's pretty good. Either way, they give you an advantage, but the middle one doesn't. So the middle one goes down, and the two over here, they are selected for. So you should be able to, if you're given a scenario, you should be able to determine if that scenario is an example of disruptive, stabilizing, or directional selection. So here we have, so what you should do is you should pause the video and then come back and check with my answer once you've tried it for yourself. And when you select an answer, explain it. Use the graph to explain it. Use the pictures to explain and justify your answer. So don't just guess. Because if you get it wrong the first time, you'll just guess the next one, and it's not really your knowledge. So actually try to reason it with the picture. So under the sea, extreme light clams and extreme dark clams are favored to the intermediate colored clams. Based on this information and the picture, determine if this is an example of disruptive, directional, or stabilizing selection. So here we have our original population. Over time, we notice the intermediates go down, and the extremes are selected for. Both of them are selected for. Our graph has been disrupted. It's been disturbed. So we have disruptive selection here. Both extremes are selected for, but the middle one is not selected for. So the intermediates were not favored in this environment. But both extremes were, so that's why we call it disruptive. Again, pause the video, try it yourself, then come back and take a look if you're right. So low birth weight babies and high birth weight babies have increased risks of complications. Thus, intermediate weight babies are favored. So you'll notice here's our original population. The extremes went down. The middle one is favored compared to the extremes. So we have an example here of stabilizing selection since the intermediate is favored. During the Industrial Revolution, trees were covered in dark ash. As a result, the dark moths were favored over the intermediate and light moths. Over time, the population shifted towards a higher frequency of dark moths. Based on this information, determine the type of selection result. So we can see our original population over here with the broken line, the dashed line. We move towards one direction only, and that's what makes this beautiful. Okay, bad joke, I apologize. So we move towards this direction, so that's directional selection. Here we have another example, and in this example here what you have to do is figure out the type of selection based on the picture that's provided of the individuals, and then you have to make a graph to illustrate that change. Again, pause the video, try it yourself, then come back and check the answer. Farmers try to protect their crops from insects by applying chemicals called pesticides. Over time, the insects without resistance die off, and the ones with resistance survive, reproduce. Eventually, insects with more resistance make up the majority of the population. Determine the type of selection and draw a graph to illustrate your choice. So, we had a mixture of some resistant and non-resistant, but over time the resistant ones are selected for. So we have a directional selection in this case because we're going really towards the resistant ones being selected for in the population. So we have a directional selection occurring. Your graph should have looked something like this. Here's your original population. On the y-axis, you have the frequency. That means basically the proportion of individuals with certain characteristics. On the x-axis, you have the phenotype written down, and then here we have the degree of that phenotype. So low resistance versus high resistance. Then the middle you have in betweens. So think of this kind of like a continuum where you have low to high and in betweens. And so our original population would have been a nice spread out curve, but then we can see the population shift, that curve shift more towards the high extreme end as well. If it was lower resistant, we would have seen our curve go towards the other end. So you should be able not to only identify, based on the graph that's shown, but also can you identify based on pictures, based on scenarios, and then provide a graph yourself to illustrate that. When you do have a graph, what I want to see is a title that's nice and descriptive. So here we can see levels of pesticide resistance. I would have even been more descriptive by saying in insects, and then frequency on the y-axis, and then like I said on the x-axis your phenotype with the spectrum shown over here. So if it was for color, it might have been light and then dark over here, and in the middle the in-betweens. So we know we're going from low to from light to dark as we go throughout. A nice broad graph to start off, and then you show that change afterwards. Usually draw your original population as a broken line, or you could choose a different color and have a legend, but it has to be specific. So I'm going to be looking for title, frequency on Y, phenotype on X, and then make sure you tell me what the phenotypes are. Have your original population and have your final population, and make sure I can distinguish between them either through a dashed line or through different colors. Either way, you should actually have a legend. So you should say dashed line like this is equal to original population. I'm just going to put OG, original, and then non-broken line would be the new population. And so this is just what you would have had to come up with to explain your choice of answers. So different types of selections have actually been demonstrated in the lab and also in real societies. A really cool example is the Tibetans. They live at higher elevation, and so we've actually seen directional selection occur there. They've been living at these higher generations for many years, thousands of years, and we've seen genes that code for their red blood cells being better at binding oxygen, and that's actually good at higher elevation because the atmosphere is thinner, so they have to be good at grasping that oxygen in order to perform things effectively. So Tibetans just have a higher, have genes that make essentially red blood cells that are better at binding oxygen. We don't have that ability. If we went to higher altitudes, we would produce more red blood cells to capture more oxygen on a temporarily, but that's not really good, a good adaptation, because it makes our blood more viscous, and that's not good for the flow, and that's not good for other aspects of your body. So really, we're seeing directional selection here for these individuals. They were selected for having a better capacity of binding oxygen for each red blood cell that they have. Some lab experiments have also shown directional selection. So for example, over here we have two graphs. Scientists had some mice, and they were trying to show that sometimes behaviors can be related to genetics. So what they did is they took mice that spent more time on their wheels running, and they bred those mice together, and they found that over time they had directionally selected for mice that could run more, and they had higher speeds as well. So they selected for the mice that just spent more time running, In the next generations, we had a higher proportion of mice that could run faster as well. So that was an example of directional selection done in the lab. So here we can see directional selection that occurred in a human population, and here we can see directional selection done in the lab specifically. Now, some might say this was an example of artificial selection. So, I mean, in reality, this was really just modeling natural selection in the lab. We weren't specifically trying to choose for higher speed. We were just trying to model natural selection in the lab. So, one interesting thought lab that I want you to work on, because it will really help you with your natural selection understanding, is trying to predict what type of selective pressure may have led to a specific trait in an organism. So, for example, if you take a look here, we have an animal trait, hawk acute vision. So what selective pressure might have led to that acute vision? Well, the ability to spot prey over long distances. If they couldn't do that, well, then they couldn't survive, eat, and reproduce. But if they could, then they would survive and reproduce and pass on their genes. Let's take a look at polar bears. So polar bears, they have white fur. So what selective pressure may have led to that? Well, if they had white fur in their white environment, they can maybe sneak up on their food more effectively and eat, therefore survive, therefore reproduce and pass on their genes. So you should be able to predict what type of environments will lead to different adaptations. So in this little challenge that you have, you're going to go through the different scenarios of adaptations and try to think of an environment that may have led to those particular adaptations. These are great examples that will help and support you to come up with answers over here. So another mechanism that could lead to evolution is the idea of non-random mating. So for a population to stay in Hardy-Weinberg equilibrium, individuals need to mate randomly. They have to do random mating. They can't be picky with who they mate with. So all individuals need to have an equal likelihood of obtaining a mate, and that just keeps the genes consistent, keeps the alleles consistent. But sometimes we have something called non-random mating, where not all individuals have the same chance of reproducing. And there's two things that can lead to non-random mating. The first we're going to discuss is something called sexual selection. Sexual selection is a special type of natural selection in which individuals choose to mate with only certain type of individuals. So here we can see the example of the pheasants. Here we have female pheasant, and here we have a male pheasant. The female pheasants are very, very picky. They choose to mate with the colorful males in the population. And so what happens is the females have sexually selected for colorful males. Even though coloration, this much coloration can be a disadvantage, these males can be eaten by predators because they're easily seen, it doesn't really matter that much because they're reproducing a lot and passing on their genes. So sexual selection is extremely powerful. It could select for things that are bad in the environment because of predators, for example, but it selects for them because, well, they get to reproduce and they get reproductive action and they pass on their genes. So these colorful individuals, they're very, very fit because they're picked by the females. And so sexual selection is an event that can lead to non-random mating, where the females are not mating randomly with everybody, they're mating specifically with the colorful males. And that sexual selection can usually lead to something called sexual dimorphism, where the males and females of a species look very different from each other. They look very contrasting from each other. So the females, they'll look very, very dull, and the males look very, 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 very colorful, and they have a lot of showy characteristics. You might have seen other examples of sexual dimorphism in other animals, for example, lions, or you might have seen it in insects, or you might have seen it in humans. Think about humans as well, where you might see sexual dimorphism. So the idea behind sexual selection is that it's similar to natural selection, except rather than the environment selecting for or against things, it's the actual reproducing individuals selecting for or against certain phenotypes. So again, sexual selection is one of the things or mechanisms that can lead to non-random mating. If there's non-random mating, Some individuals will reproduce and pass on their genes, but others just won't. So that can lead to a change in the allele frequencies based on what's selected. The interesting thing about sexual selection is that you might ask, well, how come the females aren't bright and colorful? Well, like I said, females in this case are picky. The males, they don't care what they mate with. The males will mate with whatever they see. They don't really care. So since they're not picky, there's no advantage for the females to be colorful. But there is an advantage to the males to be colorful because the females are picky. If the females were colorful, they'd probably die off because they'd be eaten by the predators, and it didn't really increase their fitness because males don't care. They'll just mate with the ones that weren't eaten with the predators, and those happen to be the peahens that are not colorful. So if the females are picky, you'll usually see more showy characteristics in the males because the males are kind of trying to compete for the females that are being very picky. So you might see some males that have more muscle mass, or some males that have larger maybe horns on their head or antlers to compete for the females. So those are all things that can lead to even more pronounced sexual dimorphism between males and females when you have sexual selection occurring. So there was an interesting experiment done just to test the idea of sexual selection. Here we have some birds with different sized tails, and what was interesting was that the male birds that had their tails shortened, they had fewer nests. They weren't mating as often. When the tails were left untreated, they had more nests, more reproductive activity compared to the short tail. Some of the birds, the scientists had modified their tail to make them longer, and they had even more reproductive action than the control one. So that was just an experiment showing that there was some non-random mating occurring in this population, where females are selecting for these longer tails in males. So males would have longer tails than the females. And so again, this is sexual selection, which is, in other words, non-random mating. You're choosing a specific mate for a specific characteristic. The females are selecting for long tail, but against short tail. And this is in the birds called widow birds. And so here you can see the result that non-random mating can have. Random mating would basically mean that if you have different, if you have variation in the population, green and blue individuals, for example, they would all mate. But in non-random mating or sexual selection, people, for example, just prefer green, so the females are always going to mate with the green ones, and as a result, the greens are just going to continue to perpetuate in the population, and the blues won't, and as a result, they may disappear from the population one day. So the thing about sexual selection is that about non-random mating is that it can often reduce genetic variation. If you're only mating with one particular type of individual, you're not going to get much variety in the offspring. Whereas random mating, if everyone is mating with everybody, well, you're going to kind of maintain that variety in your population over time. And I'm talking about a particular characteristic here. So here I'm just looking at color, for example. So random mating preserves variety, whereas non-random mating reduces that, could potentially reduce that variety, that genetic variation, because you're only choosing a specific characteristic to keep in the gene pool and not others. Another mechanism or behavior that can lead to non-random mating is artificial selection. So in sexual selection, it was individuals selecting for a certain mate. In artificial selection, we have humans choosing which individuals will mate and pass on their genes. But it's still an example of something that leads to non-random mating, because not all the individuals have equal likelihood of reproducing. Instead, for example, we might be choosing to mate turkeys or chickens with larger breasts over many generations. And so only those with the larger breast will reproduce, pass on their genes. And so we'll see a higher proportion of alleles that code for larger breast. We might see farmers that choose to breed cows for higher muscle mass, or cows that produce better milk. And so the farmers are selecting individuals to pass on genes, and as a result we have non-random mating occurring. And what I hopefully want you to get from this is that this non-random mating, in this case as well, can lead to, especially in the example I just gave here and here, can lead to some reduction in variety and diversity in populations if we're only selecting for certain types to reproduce and pass on their genes. So you can take a look at the video here to take a look at the idea of non-random mating through artificial selection. But at this point you should be able to understand the main differences between artificial selection, sexual selection, and natural selection. What I want you to get as well is that artificial selection and sexual selection are ways to lead to non-random mating, and if there's non-random mating instead of random mating, which you're supposed to have, you might have populations that evolve. Sexual selection is something that you'll see more in nature, whereas artificial selection is more human control. So if you were comparing all three types of selection, just as an aside, remember natural selection is the environment that's selecting for or against something. Artificial selection consists of humans selecting for or against something, certain phenotypes, and sexual selection would be the individual organisms of that population. For example, females selecting for or against certain phenotypes in, let's say, the males. Sometimes it could be the other way around. Sometimes in some situations you can have the males that are more choosy than the females as well. Here we can see some more examples of artificial selection that we've already discussed, so make sure you go over these and understand the examples to be able to provide them on test quizzes or during interviews or assignments. And again, there's a few videos that you can watch as you go through the PowerPoint on artificial selection and non-random mating. For example, you can see the super cow here. Another great examples of non-random mating due to artificial selection is all the different breeds of dogs. So they're all actually part of the same species, but they were created by artificial selection, by breeding certain dogs with certain characteristics, and same thing with cats. But the problem with this is that it can often lead to several health issues. So if you watch this video, you can take a look at what health issues often arise from artificial selection that humans do. Here you can see more examples of health issues that can arise because of artificial selection. Another assumption of Hardy-Weinberg equilibrium is that for populations not to change, they need to be large. If populations are small, they can go through something called genetic drift. And so genetic drift is when allele frequencies change due to chance. So they randomly change due to chance. And genetic drift is more likely in populations that are small. And the reason that they're more likely in populations that are small is because when a population is small, one change can be often devastating in the next generation. If I have a small population of flowers over here, for example, and let's just say, and here we have 10 flowers, it's not a lot. I'm walking over the flowers, I love flowers, but some of these flowers were in the wrong place at the wrong time, or some of the flowers, just by chance, they didn't make good seeds. So what happened was that the only one that made good seeds was maybe this one over here. And because only this one made good seeds, the rest of these ones died off. This one over here and this one over here, they made good seeds. Because they made good seeds, they reproduced, passed on their genes. The rest didn't make good seeds, and so they did not pass on their genes. They just died off. And so what happened was we lost little r allele. If this population was larger, there might have been a larger chance that other organisms or other flowers could have reproduced and passed on their genes, but we lost these flowers here because the population was small. Maybe they didn't produce good seeds, maybe somebody stepped on them, they were in the wrong place at the wrong time. It's just due to chance we lost that allele. So in a small population, it's a devastating effect to lose certain individuals, and it will change the allele frequencies. if I have a large population of flowers and I step on one flower, yeah, it's sad, I lost a flower, but there are other white flowers there that can maybe pass on a little R allele, and I won't even notice it's gone. So genetic drift is really a change in the population due to random chance. The surviving individuals didn't necessarily survive because they were better or more fit for the environment. It was just that they survived. And so if you have a small population, changes are more likely to happen in that population that'll change allele frequencies. I'm not saying that allele frequencies can't change in large populations. They can, but it may not be significant. Like I said, losing one white flower is not going to be a big deal if you have thousands of flowers in that environment. I'll talk about this example a few times, that it's like having a test out of 2. If you have a test out of 2 and you lose 1 mark, that's already 50%. If you have a test out of 100 and you lose 1 mark, you're at 99%. No big deal. We barely even notice it. So the higher that population of that test mark, the less losing 1 mark will be the less of an effect losing 1 mark will have on your overall representation, as opposed to if something's out of 2 and you get 50%, it has a big effect on that mark overall. So with genetic drift, the important thing to understand is that small populations are more likely to experience that random change compared to large populations. There's 2 processes that can lead to genetic drift. There's something called the bottleneck effect and the founder effect. So these 2 processes essentially can reduce population sizes, and as a result of reducing population sizes, genetic drift will occur, which means a change in the allele frequencies due to random chance, and it's really higher in the smaller populations compared to large populations. So here we have just an example again of a small population. There's a lot of variety, but it's a small population. And by chance, just randomly, these 3 brown frogs, they die. Now, if this was a large population where there were a lot more individuals, if 3 brown frogs died, I mean, yeah, it's sad, death, okay, but maybe we wouldn't have noticed it because other brown frogs might have reproduced and passed on their genes. But in a small population, maybe these were the only ones carrying the little r, or whatever it was. And so now we have no more. And so that led to a change in our gene frequencies. The little r is completely gone from our population, and so that led to evolution. So this is an example of genetic drift, a change in gene frequencies due to chance. They randomly died. It might not have been natural selection. They didn't make it through. They didn't make it through. Someone stepped on them. They were in the wrong place at the wrong time. It had nothing to do with their environment, or could have, but small population, and the change was random. But we really did see the effect because it was such a small population. So what can lead to genetic drift? Like I said, there's 2 things. There's the bottleneck effect and the founder effect. So the bottleneck effect is when you have a large population to begin with, but then some drastic event occurs and the population shrinks down the size. So there might have been a drought, there might have been an earthquake, there might have been a flood, there might have been humans hunting and killing a bunch of individuals off. And so what happens is only a few individuals survive. And what I want you to notice is this. In our large population, we have all these different color marbles here that represents the variation that existed. After the bottleneck event, so whether it was overhunting or whether it was a flood, whatever it be the case, this is our surviving population. Notice that there's not a lot of, we lost some variation. We don't have those yellow ones anymore. So when this population eventually recovers, you'll notice that there's less variation because our surviving population just had less variation to begin with. And this surviving population is small. If that green one had died, and some of the ones carrying the little green allele didn't survive or reproduce, then we could have run the risk of losing the green one too and only having purple. So the main point I want you to get here is that the bottleneck effect is reducing the population size. And when you reduce the population size, you have a chance of losing your diversity. And your allele frequencies can change based on random events more easily than if your population size was large. We call this the bottleneck effect because it's kind of like trying to pour these marbles out of a bottle. The bottleneck is narrow, and so not a lot of those marbles will get out. Only a few will. And that's our surviving population, the ones that got out, but not many did. So bottleneck effect can lead to genetic drift because it really reduces the size of the gene pool. And what often happens is that some individuals are overrepresented in the surviving population, and some individuals are underrepresented in the surviving population, like this green one here. And some individuals don't show up at all, like this yellow one over here. And so that reduces diversity, and it's not good for a population. You need to have variation for survival of the species. So this actually happened. I mentioned hunting was an example of a bottleneck event, where cheetahs were being overhunted. They were being shot like crazy, killed in many ways, and only a few of them survived. So they had a very low population. Their population is starting to recover, but what we noticed is that that population has very low genetic diversity because the ones that survived just happen to be very similar to each other. And so whatever variation we had to begin with in the initial population was shot out or overhunted out, and so our new population really has very little diversity left over. So again, here we can see a bottleneck event. In this example, we had a good variety of organisms to begin with. There was some kind of flood or something that occurred, and only a few individuals survived. Now here you'll notice that none of the individuals of Big B survived. It was just they did not have the opportunity to survive. Our population is much smaller now, and it's very, very easy to either go extinct, or when this population does grow, there's going to be very, very little variation left in the population. The next event that can lead to genetic drift is something called a founder effect. So remember that genetic drift is a change in the gene pool due to random chance, and that genetic drift is more likely to happen when your population is small compared to large. So the idea with the founder effect is you have a large population to begin with, and some of the individuals go to find a new area to live. So I found a new area to live, founder effect. So these individuals here and these individuals here, they went to different islands to live on. And what I want you to notice is that those individuals are not necessarily representative of the variation that existed in the original population. So as the founder populations increase in size, you'll notice that they have some different frequencies compared to the founding populations that they were made from. And they even have different frequencies compared to the original population that the founders came from. So for example, over here, you'll notice that we have fewer green than before. Over here, you might notice that we might have fewer of these intermediate, these lighter brown ones than before, compared to the original population. So what happens is, in the founder effect, you have some individuals from a large population, they go and they find a new environment. They're a small population, and because they're a small population, some random chance event could lead them to looking drastically different. There might be some underrepresentation. Same idea could happen here. You might have, maybe if this one died off, maybe you would not have seen many of the green ones in the next generation. And you can kind of see that here. There's not a lot of green ones left in that generation. If this one died off, you might not have seen any at all in that generation. Same thing with this one here. So the founder effect is in a way similar to the founder effect, similar to the bottleneck effect. It's just that rather than being a drastic event that led to a reduction in population size, it's just some individuals from a larger population decide to move, and that's all it is. So the idea is that genetic drift, small population sizes can change due to chance more likely than a large population changing due to chance. The things that can lead to a small population size and therefore increase the chance of genetic drift are the bottleneck effect, where there is a catastrophe, overhunting, flooding, or the founder effect, where a small subset of individuals from a large population go live somewhere else. And because they're a small subset, they might just be less variety than the larger population that they came from. So over here we can see the founder effect. We had a large population in our mother population, our original population, and then there might have been a few individuals that decide to go live somewhere else, and they do not represent the variation that the original population represented. You'll notice that there was only one red one here. So this is kind of scary, because if this red one goes, and some of the ones that carry the red alleles, if they don't reproduce in a small population, that could be devastating. That could mean the red ones don't exist anymore. Here you have just a lot more red, a lot more yellow. You have a larger population size. So if you kill one red one, yeah, it's sad, but you might not even notice. If you kill this one, that's pretty bad for the allele frequency. There's not many individuals anyways to begin with. Think of it kind of like a test that's out of 2 marks. If you get one wrong, you're at 50%. Think of a test that's out of 100 marks. If you get one wrong, you're at 99%. So one question wrong out of 100 marks, no big deal. One question wrong out of 2 marks, you lose 50%. So small population, small changes, random changes are more noticeable. And so the idea is that the founder effect and genetic drift and the bottleneck effect and genetic drift, they lead to a loss, they could lead to a loss of genetic variation. Here's an excellent example that happened in human populations. So there's a disease called Huntington's disease. You might have heard about it in one of our previous presentations or from your readings. So what happened was there was a subset of individuals from the original population that went to live somewhere else, and these are called this is called the founder population over here. In this original population, the Huntington's disease was very, very low frequency. One individual out of all these individuals here had it. But now look at our founder population. Yes, it's still only one individual, but it's one individual out of 1, 2, 3, 4, 5. 1 out of 5 individuals have this disease now. Whereas 1 out of, I don't know how many there are here, but 1 out of this many people over here is not that much over here in this case. Now our founding population, as the individuals reproduce, because the population is so small here, this Huntington's individual might have reproduced more frequently, and now as the population grew, we had more individuals with Huntington's disease, because this individual was just more represented compared to where it came from the original population. So this is a great example of the founder effect leading to a change in allele frequencies over time. Over here we can see 2 graphs comparing different size populations and their change in allele frequencies. Here we're keeping track of the big A allele. And I want you to notice the frequency. So we have frequency of allele A, frequency of allele A over here. But what's different is the population size as well. So here we have 25 stoneflies, and here we have 500 stoneflies at the start of every generation. And so we're not really applying any selective pressures. We're just analyzing the populations over a few generations. And what we noticed is that if you keep track of the gene frequencies over time, in the small population, you have drastic changes. You can see here, this is where we're sort of starting off generation 1, and then in 5 populations, the big A went up, but in 4 populations, the big A went down. So there was drastic changes there in the frequency of big A in that population. But if you look at the large population, yes, there was some fluctuation that happened compared to where it started off, but it was nowhere near as drastic as what you saw in the one with the small population. So the idea is that small population, some random chance led to huge changes in an allele frequency compared to the large population. Random chance led to some changes, but not much because it was such a large population. So Here you can see allele A neither lost nor fixed, and so what I mean by that is, in this example, these ones, allele A was lost. In this example, allele A was fixed. Over here, allele A just stays stable. No significant changes in allele A in the larger population. So the key point to remember is that drift and natural selection, they're both things that can lead to evolution. They're both mechanisms of evolution, but drift is really more random. The surviving individuals are not necessarily more fit for the environment. It was just a random event that led to some individuals dying and others surviving. So you're walking through a path and there's some insects and you're in a rush and the green insects just happened to be sitting here at the time that you were walking through it. They died off and not many survived, so the next generation has more brown insects. And so drift again is more random, and this is more pronounced in a smaller population. Because this population is so small, losing 3 of the brown insects, those are 3 of the green insects, these are the only green insects in the population, is devastating. We're losing a lot of those green alleles, especially if these individuals don't have the green alleles in them. If the population was large and there were thousands of insects and we lost 3 green insects, no big deal. That change in allele frequency would not have been very noticeable. If you think about it again on a percentage scale, like on a test that we saw, 1 out of 2 versus 99 out of 100. So drift doesn't lead to adaptations. That's the big idea to get here. The individuals that survived, they didn't survive because brown is necessarily better in this environment. It's just that they survived because they were sitting over here as opposed to up here and didn't get stepped on. So it was just really, we wouldn't call this an adaptation in this situation. It's just a variation that just happened to survive by accident. The next condition in Hardy-Weinberg equilibrium is that there can't be gene flow. Basically, you can't have immigration or emigration. So gene flow just means the exchange of genes with another population. So you have 2 different populations that are kind of separated, and what happens is, so here we have the green and the brown, and here you can see a brown insect traveling to the green insect area, and it might start mating. So initially there might not have been any brown insects in this population, but now that we have some gene flow, a brown individual going into the green area, those brown genes might be introduced into this population, and so this population of future generations may have more brown genes than it had before. And so we might see some evolution happen there as a result of that. So gene flow can introduce new alleles into a population. And so what usually happens is when gene flow happens, it actually reduces variation between the populations. I don't necessarily mean within a population, I mean between. So for example, here there were all brown, here there were all green. These populations were drastically different from each other. Now if the greens start to move here and the browns start to move here, then what's going to happen is they're going to start mating with each other, and so these populations won't be so different after all. They're going to start being more similar to each other, and they might even start living closer to each other. Who knows? But the main idea is that when you have gene flow, you have new alleles being introduced into a population, and that could lead to changes over time. The last condition for Hardy-Weinberg equilibrium is the idea that there can't be mutations, because mutations introduce new alleles into populations. So we saw this example to begin with. You only had green, mutation happened, now you have a brown allele, and that could lead to allele frequency changes over time, which could lead to evolution. Now mutations are typically rare, especially ones that lead to drastic changes in phenotypes. So the chances that a mutation alone is going to lead to evolution is unlikely. However, if you combine mutation and natural selection together, then that could lead to evolution. So usually you have to see a mutation happen, and then natural selection might select for that new phenotype, and then that could lead to a more drastic change in the allele frequency over time. So you can see the example over here. Here we have some mutations that led to maybe some antibiotic resistance in bacteria, and there's not many antibiotic resistant bacteria in there at the moment, so mutation alone doesn't lead to much change. But now once we add the antibiotic in, we see that there's only the resistant bacteria that are left over time. So there's a drastic change that occurred there, and that's because we combined our evolution, our mutation, with the power of natural selection. So those were all the mechanisms that lead to evolution. Of all the mechanisms that we saw, the ones that are more powerful and that are more likely to cause evolutionary change are natural selection, genetic drift, and gene flow. Um, the other ones can cause change too, but the ones that are more powerful are these three here. The interesting thing is, of all of those mechanisms of evolution, the one that consistently leads to adaptive evolution is natural selection. Adaptive evolution just means that the new population, the way it looks, is just that it's better fit for its environment. Genetic drift and gene flow, they're more, let's say, random. The population will just change because new alleles came in to mate, or individuals just died off randomly, not because of the environmental factors necessarily. But natural selection is really because of the environmental factors that certain individuals died and certain individuals reproduced. So natural selection consistently leads to adaptive evolution. Maybe genetic drift and gene flow might, but that's rare. It's natural selection that consistently does that. So the other two, like I said, it's mostly a chance change, just some random change. So again, natural selection all depends on the environment. Big idea in this course. This is a summary table of the different mechanisms of evolution and the effect that they might have on variation. So you should understand that the different mechanisms could either increase or preserve variation, or maybe even decrease it. If you look at natural selection, what does it do to variation? Well, it could preserve it or increase it if you have disruptive selection, but it could also decrease variation if the type of selection that occurred was a directional selection where only one of the extremes, or if it was a stabilizing selection where only the intermediates are selected for. So natural selection has the power to either increase or preserve, or even decrease variation. You should understand which situation that can occur. Non-random mating, does it increase or preserve variation? It doesn't really increase variation. It actually could decrease it or prevent it from going up. And that's because only select individuals reproduce. It's not randomly mating anymore. Gene flow, within a population, within one population, it will increase some variation. But between populations, it'll actually decrease variation. So populations, when they exchange genes, they become more similar to each other. It makes sense because now they're all mating with each other and sharing their genes instead of keeping them a secret. When everyone knows the secret, now you all share something in common. Genetic drift, that often reduces variation. It doesn't increase variation. So a lot of alleles could be lost completely when your populations are small and some random change happens. Mutation, typically that's going to increase variation. So that leads to new alleles coming into the population. It won't, some students might say, well, doesn't mutation decrease variation if you have natural selection happening and one mutation selected for as opposed to the other? Yeah, but that's natural selection doing that. So mutation by itself won't necessarily decrease variation. Mutation will typically increase the variation. So here we can see the summary of the assumptions of the Hardy-Weinberg equilibrium. In order for a population to be in the HWE, in other words, in order for a population not to evolve, there cannot be selection, there cannot be mutations, there cannot be migration or gene flow, the populations need to be large, and the populations need to be randomly mating. If there's selection, if there's mutation with combination of maybe selection, if there's some gene flow happening, if there's small populations, and if random mating is not occurring but there's non-random mating due to sexual selection or artificial selection, then populations may evolve and therefore they won't be in Hardy-Weinberg equilibrium anymore. So now we're kind of summarizing what we've seen throughout this unit so far. So we learned that individuals do not evolve, it's the populations that evolve. Natural selection is just one mechanism that can lead to differential reproductive success. Remember that the population is the smallest unit that can evolve. That's super important to remember. It's not the individuals themselves that evolve. And so what we saw is that microevolution is a change in allele frequencies over time, from generation to generation, and they can be caused by those 5 mechanisms that we discussed in this presentation. A great example that you should read up on, there's also a great gizmo on this that some of you might have chosen to do for your assessment. If you didn't do the gizmo, it's a good idea to try this one too. It's a really great visual of understanding evolution. So I want you to read through this example. Essentially what scientists noticed is that during drought, the food that was left over for birds had a hard casing that only birds with large depth size could actually crack and eat. And so what we noticed is that during periods of drought, we saw average beak sizes increase a little bit. So we saw some directional selection occurring. And what you need to understand, hopefully you get this now, is that the small birds were not transforming into, small beak birds were not transforming into big beak birds. It's just that the small beak birds were dying off. They couldn't survive, and they couldn't reproduce. The big beak birds, they could survive, they could reproduce, they could pass down their genes, and that's why you have a larger average beak size in these situations here. So if you can understand that idea, you're really starting to understand the idea of evolution. So evolution is the change in the population, the allele frequencies. So we might have had a variation before, or our average was about a beak size of 2.5, but then over time the average became a beak size of 3.5. That's our evolution. The mechanism that led to this was natural selection in this case. And so this is the example of the Darwin finches. It's a great idea to read about this example in your textbook and to be able to explain this during an interview, test, quiz, or other assessment. And so we started off with this slide, and hopefully now it makes a lot more sense to you. If you can't explain it to yourself, go back and review this PowerPoint, review the initial PowerPoint, and try to make that connection between why we started off with this slide and why we're ending off with this slide. One important concept I do want you to read up on is the selective pressures that humans can provide, either through artificial selection, but also by accident sometimes, and unintended circumstances that we might have. So you should know at least 2 examples from this table here. It's also in your textbook. There's more examples that you can look at of a human selective pressure and the evolutionary change or consequence that it had for organisms in the environment. Try to understand maybe what mechanism of evolution occurred and why that selective pressure led to an evolutionary adaptation. So again, take a look at this. You should know at least 2 great examples of how humans can lead to that evolutionary change in populations. These are some great practice questions for you to attempt based on this PowerPoint from the textbook. There's also a few videos that you should watch to help you understand these concepts in a bit more detail if you need some more support. There are some practice questions designed for this PowerPoint specifically, so you can try those. The Nelson readings for Chapter 8 are great to do so you can get ahead of the course. And then if you don't understand evolution and need a bit more support, there's some great videos online, but also this website here, evolution.berkeley.edu, they provide an excellent resource to explain these evolution concepts in fantastic detail, but also they keep it nice and simple. So I recommend that you check that out if you need a bit more support for evolution.